Alright, what's up guys? So today is the third installment in our protein series, and since you're all amino acid experts now, we're going to start combining these amino acids together to form dipeptides, oligopeptides, polypeptides, and fully functioning proteins. So let's start simple by just combining two amino acids together to form a dipeptide. So we're going to connect amino acids together by what is called a peptide bond, and this is catalyzed by the large ribosomal subunit. So we'll talk about this a little bit more when we get to nucleic acids, but the ribosome is a non-membranous organelle. So most organelles have a phospholipid membrane. Ribosomes do not. They are strictly RNA and protein, and they are broken up into a small component or a small subunit and a large component or large ribosomal subunit. So take a look at this reaction here on the right. So we're joining together two amino acids to form a dipeptide, and we're losing water. So when in doubt, lose water. So this is a dehydration or a condensation reaction. So what type of functional group is being formed here? So go ahead and pause the video and see if you can figure out what new functional group we have created. So the peptide bond that we created is an amide. So the peptide bond has what we refer to as a partial double bond character. So where have we seen this before? What does that mean? So this is similar to what we saw when we talked about aromatic rings earlier in the semester. So just like how all of the carbon-carbon bonds in a benzene ring are equivalent, remember we talked about, imagine how a benzene ring is constructed by 6 carbons that are single bonded to each other, but we have 6 additional electrons that flow throughout the ring, making all carbon-carbon bonds in a benzene equivalent 1.5 bonds. Well, we see the same thing with the peptide bond that forms when we join two amino acids together. So the CO bond and the CN bond are equivalent. They're equivalent 1.5 bonds. Those 2 electrons are able to flow between these 3 atoms. So this is going to create a rigid structure. So both the CO bond and the CN bond are going to be rigid. You're not going to be able to rotate them. And so whenever we have something rigid that can't be rotated, if we have substituents coming off of both sides, we're going to get geometric isomers. And when we're talking about proteins, when we're talking about peptide bonds, they almost always exist as the trans isomer. Okay, so let's learn how to draw and name small peptides. So whether you're drawing or naming, we're always going to start with what we call the N terminus. So the N terminus, N for the nitrogen in the amine group, is the end with the unreacted amino group. So that means you're going to end with what we call the C terminus. So that's the end with the free carboxyl group, C for carbon. And you generally are going to alternate R groups going up and down, and we're going to write names with 1-letter codes or 3-letter codes separated by hyphens, starting from the N terminus all the way to the C terminus. Okay, so let's try one. So what is the name of this oligopeptide using 1-letter codes? So I want you to pause the video, look at your amino acid handout, and see if you can identify all 5 amino acid residues in this oligopeptide. And then I want you to list their 1-letter codes in order from N terminus to C terminus. Okay, so it looks like this first amino acid is alanine, followed by glycine, serine, valine, glycine. So if you wrote AGSVG, you are correct. So go ahead and pause this video again and see if you can do the same thing, but using 3-letter codes. Okay, so we have ALA, ALA for alanine, GLY, gly for glycine, SER, ser for serine, VAL, val for valine, and GLY, gly for glycine. Okay, so just like we can calculate the isoelectric point of a single amino acid, we can do it for a longer polypeptide or oligopeptide chain. So it's going to have similar steps. So what we're going to do is draw the peptide using what I call shorthand, and I'm going to show you that in the next slide. We're going to start at pH 1 just like we did with amino acids. We're going to label each titratable group with the pKa, move up from pH 1 until we reach the first pKa, Draw a reversible reaction arrow and write the lowest pKa on top of the arrow. Draw a new structure with the correct H plus removed. And then we're going to repeat this until we remove every proton that can be removed. Then I want you to identify the net charge for each species, find the species with 0 net charge, and just like with amino acids, our PI for our peptide chain is going to be the average of the pKa you wrote above the reaction arrow on both sides of the neutral species. Okay, so what we're going to do is calculate the PI of FKEDKSA. So we're going to start with a shorthand drawing. So what this means is we're going to write the codes for each amino acid connected with bonds. So just hyphens between them, and those are going to be our peptide bonds between each amino acid residue. Okay, I'm going to get you started here by writing the shorthand for our oligopeptide. And then for homework, I want you to finish this up and see if you can come up with the PI for this oligopeptide. So we're going to start with the N-terminus, and our N-terminus at pH 1 is going to be protonated. Our first residue is a phenylalanine, followed by lysine, glutamic acid, aspartic acid, another lysine, serine, alanine, and then our C-terminus. Okay, so now we're going to add in our titratable side chains. So we already have our titratable N-terminus and our titratable C-terminus. Remember, we don't have any other N or C termini now because they're all caught up in these peptide bonds here, right? But like I said, we do still have to worry about our titratable side chains. So phenylalanine does not have a side chain pKa listed, so we don't have to worry about its side chain. Lysine, we have an amino group on the side chain, which is titratable. Glutamic acid has a carboxylic acid side chain, which is titratable. Same thing for aspartic acid. Same thing for our other lysine. And serine and alanine, neither of those have a third pKa value, so neither of those side chains can be titratable. Okay, so go ahead and see if you can finish this problem out and come up with a PI, and then we'll go over it together on the next video.